Every day, yet I said to thy conquer to Holy Word. Father, your Son, Holy Spirit. Amen.
Good morning, everyone. We are Group 2, and today we will be presenting our video report about types of cells, cell structure, and function.
Basic types of cell. Cells comes in a variety of shapes and sizes, but all cells share some basic characteristics. One thing that all cells have in common is a plasma cell membrane. They have in common because they provide protection for a cell. The cell membrane is a boundary which allows things into and out of the cell. Differences in cell types. All cells fall into one of two categories, the eukaryotes and prokaryotes. When we say eukaryote cells with a membrane-bound nucleus and membrane-bound organelles, when prokaryote cells without a membrane-bound nucleus and membrane-bound organelles. Nucleus is the central organelle of a cell that contains the genetic material DNA. The nucleus serves both as the repository of genetic information and as the cell's control center. Organelles are like organs for the cell. They are special structures that perform vital functions necessary to the cell, or they perform various jobs inside the cell. Differences in cell types. Prokaryotes in bacteria and eukaryotes in animal and plants.
Prokaryotes, nucleoid cell wall in the sum of eukaryotes, and axon, sum of prokaryotes, flagellum, ribosome, cell wall, eukaryotes, say nucleus, nuclear membrane, ang pareha sila may cell brain o endoplasmic reticulum o say cytoplasmic membrane, chloroplast, Golgi, mitochondrion o cytoplasm. Next, traits of eukaryotes. They are have a nucleus where the genetic materials of the cell of store. Number 2, they have many organelles that work together to help the cell function. Number 3, eukaryotic cells are much more complex than prokaryotic cells. Number 4, they can be just one cell or can make up more complex multicellular organisms. Number 5, all plants, animals, fungi, and protists are eukaryotic cells. Next, traits of prokaryotes. They do not have a nucleus and their genetic materials is not stored in the nucleus. Number 2. So they have some organelles structures but not many. Number 3. They are less complicated than eukaryotes. Number 4. All bacteria are prokaryotes. 5. Most are unicellular, but some prokaryotes are multicellular. Thank you.
So we have prokaryotes differ from eukaryotes in several ways, including but not limited to. It means prokaryotes are always unicellular, while eukaryotes are often multi-celled organisms. In addition, eukaryotic cells are more than 100 to 10,000 times larger than prokaryotic cells and are much more complex. The DNA in eukaryotics is stored within the nucleus, while DNA is stored in the cytoplasm of prokaryotes. As you can see, we have the characteristic prokaryotic and eukaryotic. Next is we have organelles in common. Both cells contain the following structures like cell wall, cell membrane, ribosomes, cytoplasm, and etc. And so we have cell wall, the firm non-living layer that encloses and supports the cell of most plants like bacteria, fungi, and algae. In plant cell, the cell wall support and protect the cell. The cell wall shapes, supports, and protect the cell wall or to prevent the cell from absorbing too much water and bursting. Cell wall are rigid and unbending. The apparent rigidity of the cell wall, like for example inflation, this inflation is result of the passive uptake of water. In plants, a secondary cell wall is a thicker additional layer of cellulose which increases wall rigidity. Plant cell support itself. An animal cell does not have a cell wall because plant cell have a cell wall like chloroplast and other specialized plastids and a large central vacuole, which are not found within
Cell membrane. Cell membrane structure component arrangement. Cell membrane have a two parts, eukaryote, prokaryotes. For eukaryotes, membrane includes nucleus, nucleolus, mitochondrion, ribosome. For prokaryotes, ribosome, nucleus, capsule, flagellum, cell wall. Function include barrier, transport, now diffusion, osmosis, facilitated diffusion and active transport. Recognition, e.g. self versus non-self. Reception, for protein harmonies and last adhesion. The cell membrane with its distinct components and function, it is a fundamental element in cell structure and function, facilitating communication, transport, and maintaining cellular integrity in both eukaryotic and prokaryotic cells. Ribosome. In structure, RNA, protein. Function, site of protein formation. Found in both prokaryotes and eukaryotes. Different structural. Pre-ribosome. Ribosome associated with the root endoplasmic reticulum. Ribosome are the cell's protein factories. Ribosome composed of rRNA and protein serve as the key cellular machinery for protein synthesis. They operate as pre-entities in the cytoplasm or associated with the root endoplasmic reticulum. Contribute to the diverse protein need of the cells. Ribosomes are versatile structures found in both prokaryotic and eukaryotic cells with variation in their structure between the two cell types. Cytoplasm. The jelly in the center. Cytoplasm in the cells is jelly-like liquid that fills all the empty space in the cells. It is also known as protoplasm, which is the dense gelatinous solution within the cell membrane that is the primary site of the cell's biochemical and synthetic processes. Cytoplasm often refers to as the jelly in the center. The protoplasm is a gel-like substance filling the cell's interior, serving as the primary location for essential biochemical and synthetic processes.
Vacuole is a membrane-bound cells organelles. Both animals and plants have vacuole. In animal cells, vacuole generally small and help sequester waste products, and in plant cells, vacuole help maintain water balance. Nucleus is the membrane-enclosed organelle within a cell that contains chromosomes. There are parts of nucleus and its structures and functions. Nuclear envelope structure: the largest cell organelle covered with a thin double-layered nuclear membrane. Its function: regulates cell function, including metabolic and chemical activities. If removed, the cell dies. Nucleus structure: most spherical, dense structure containing jelly-like fluid called nucleoplasm. Function: contains chromosomes which bear genes responsible for hereditary characters. Plays a key role in cellular reproduction. Nucleoplasm structure: it is the jelly-like fluid within the nucleus. Function: surrounds the chromatin fibers and nucleus, providing support to the organelle. Endoplasmic reticulum structure: network of thread-like structures called chromatin fibers containing DNA and nucleus. Function: it helps in the formation of ribosomes. Plays a role in cellular reproduction. Chromatin structure: thread-like structures containing genetic material. Function: bears genes that carry hereditary information involved in the information of ribosomes. This information outlines the roles and structures of these components within the nucleus, highlighting their significance in regulating cell activities, hereditary information, and cellular reproduction. Nucleoid region in prokaryotes. The nucleoid is the region in the cytoplasm where the chromosome DNA is located. The DNA in the nucleoid is circular in prokaryotes. Unlike eukaryotes, prokaryotes do not have a membrane-bound nucleus. The nucleoid is not a distinct organelle but rather an area of an cytoplasm where the strands of DNA are concentrated. In summary, the nucleoid region serves as the location for the genetic material in prokaryotic cells. It lacks the membrane-bound structure seen in eukaryotic cells, highlighting one of the key differences between prokaryotic and eukaryotic cells organization.
So, what is endoplasmic reticulum or structure and function? So, the endoplasmic reticulum or ER is a large dynamic structure that serves many roles in the cell including calcium storage, protein synthesis, and a lipid metabolism. The diverse function of the ER are performed by distinct domains, consisting of tubules, sheath, and the nuclear envelope several proteins that contribute to the overall architecture and dynamics of the ER have been identified, but many questions remain as to how the ER change shape in response to cellular cause, cell types, cell cycle states, and during development of the organisms. Here we discuss what is known about the dynamic of the ER, what questions remains, and how coordinated response add to the layers of regulation in these dynamic organelles. So next topic, smooth versus rough endoplasmic reticulum. So what is smooth ER? Smooth ER is a largely associated with lipid fat manufactured and metabolism and steroids production, hormone production. It also has a detoxification function. The next, what is Roth ER? Roth ER, studied with millions of membrane-bound ribosomes, is involved with the production, folding, quality control, and dispatch of some proteins. So, what is a ribosomes? What is the function of ribosomes? A ribosomes is an intercellular structure made of both RNA and protein, and it is the site of protein synthesis in the cell. The ribosome reads the messenger RNA or mRNA sequence and translates that genetic code into a specified string of amino acids which grow into long chain that fold to form proteins. So, function of nuclear envelope. The nuclear envelope separates the content of the nucleus from the cytoplasm and provides the structural framework of the nucleus. The nuclear membrane acting as a barrier that prevent the free passage of molecules between the nucleus and the cytoplasm maintain the nucleus as a distinct biochemical compartment.
Golgi apparatus, the structure and function. So first, structure. These are very small vesicles which occur in different shapes such as granules, filament, or rods. So first function, it is concerned with the synthesis and secretions of enzymes and hormones in the cell. Second structure, it consists of many small groups of hollow tubular structures with membranous walls and is associated with some minute vesicles and vacuoles. Second function, Golgi apparatus carries material synthesized by the endoplasmic reticulum to different parts of the cell. The material is stored and packaged in vesicles. Third structure, these vesicles are arranged parallel in stacks called cisterns. Third function, it forms glycoproteins and complex sugar GA-lactose from simple sugar. Fourth structure, the internal space of the vesicle is called the lumen. So, gitawag ang lumen in internal space of the vesicle. So, fourth function, it also forms a chromosome of sperm, only in animals and human beings. Peroxisome is a carry out oxidation reactions that break down fatty acids and amino acids and detoxify poisons. Mitochondrion. So mitochondrion is a production of ATP and enclosed by the inner membrane is a jelly-like matrix. And folds are called cristae. And napo ni sila ilahang kaugalingon nga kuan DNA.
My topic is chloroplasts and lysosomers. So, chloroplast. The chloroplast is an organelle within the cells of a plant. So, this is the structure of chloroplast. Have the outer membrane, inner membrane, granum, lumen or stroma, and thylakoids. Ang chloroplast has a pigment containing. Pigment containing is can absorb light energy and has a small flattered sacs called thylakoids. Thylakoids is a transport of proteins help to absorb the sunlight. And page 24 is the lysosomers. For intracellular digestions and suicide bag of the cells. So mao ni siya lysosomers. Ang iyaha day siya ang function is to digest and kani siya nang to digest food also sa mga broken organelles. So diri sa 25 makita nato ang lysosomers nga ay mao ni siya functions and structure sa lysosomers. Iyang function is to digest food. used to make energy. So, ang usapod kay is to clean up and recycles the digest broken organelles. So, mao ni siya lysosomers, no? Nga mao ayang function is to mo digest siya og foods to make an protein. And also mao ni siya diri sa pikas, mao ni siya agianan sa mga broken organelles. Diri mo agi to mga broke nga mga organelles ug mao ni siya structures sa lysosomers. Structures membrane sacs of digest enzymes. So mao ni siya nga lysosomers release digestive enzymes to digest all cells, and this is to lead the death of the cells.
Today, I will gonna tackle about microtubules, flagella and cilia, and centrioles. First, microtubules. Microtubules are dynamic structures within cells that play essential roles in various cellular processes. They are long, hollow cylindrical structures made up of protein subunit called tubulin. Microtubules are the critical component of the cytoskeleton which provide structural support to cells, and it's involved in cell divisions, cell shape, intracellular transport, and cell motility. The primary function of microtubules to provide structural support and maintain cell shape. They form a network of tracts throughout the cells serving as roads along which organelles, vesicles, and other cellular components can move. Microtubules also play a crucial role in cell division by forming the mitotic spindles, which help separate the duplicated chromosomes into two daughter cells during mitosis. In addition to their structure's functions, microtubules are involved in intracellular transport. They act as tracks for motor proteins such as kineses and dynein, which carry cargo along the microtubules. This transport system allows molecules and organelles to move the specific location within the cell. Microtubules are also important in cell motility. In some cells, such as sperm cells and certain types of immune cells, microtubules form the core of structures called flagella or cilia. The beating motion of these structures enables cells movement or the movement of fluids over the cell surface. Microtubule also has protein subunit called tubulin. Tubulin, a globular protein that polymerize the form microtubules. Microtubules are microscopic hollow tubes made of protein called alpha and beta tubulin. They are crucial component of the cytoskeleton, which is a network of protein filaments that provide structures and shape to eukaryotic cells. Microtubules play a role in various cellular processes, including cell division, intracellular transport, and cell movement. They are involved in organizing the cytoskeleton and transporting materials within the cell. Second, flagella and cilia. Flagella and cilia are both structures found in cells that are involved in self-movement. They have a similar structures but differ in their size and function. Flagella are long whip-like structures that extend from the surface of certain cells. They are typically found in single or small number of cells and are used for locomotion. Flagella can be found in various organisms, including bacteria, archaea, and eukaryotic cells such as sperm cells. The movement of flagella allows the cells to swim proper themselves throughout environment. Cilia, on the other hand, are shorter and more numerous compared of flagella. They are hair-like structures that extend from the surface of cells. Cilia are found in large number of the surface of many types of cells, including those lining the respiratory tract, fallopian tubes, and surface of certain cells in human body. The coordinated movement of cilia helps in moving fluids or particles along the surface of the cells or in creating a fluid over the cell surface. Both flagella and cilia are composed of microtubules, which are cylindrical structures made of protein called tubulin. The microtubules provide the structural support and enable the movement of flagella and cilia. Last, centrioles are small cylindrical structures found in animal cells. They are composed of microtubules and are typically found in pair near the nucleus of the cells. Within a region called the centrosome, centrioles play an important role in cell division and the organization of the cytoskeleton. Centrioles are involved in organizing microtubules in the cytoplasm. So that was microtubules, flagella and cilia, and centrioles.
Centrioles organizing microtubules that serves as the cell's skeletal system. Microtubules stabilize and maintain the centrioles through the cell division. Centrioles has 9 plus microtubular arrangement and 4 cellular division. Nucleolus, a condensed region of chromatin where ribosome synthesis occurs.
Types of cell structure and function.
So this is the Venn diagram of prokaryotic and eukaryotic. There are differences and similarities. So first, prokaryotic. We have nucleoid, cellular DNA that floats freely around cell, simpler DNA, no membrane-bound organelles, typically much smaller. Divide by binary fission, typically uncellular, and anaerobic and aerobic. So next is prokaryotic. We have nucleus, linear DNA that is held in the nucleus, complex DNA, membrane-bound organelles, typically much larger. Divide by mitosis and meiosis, typically multicellular and aerobic. So, next is their similarities. We have chromosomes, DNA as genetic material, ribosomes, cytoplasm, plasma, membrane, sometimes have cell walls, and the last is
Next is the plant cell and the animal cell. So first, the parts of the plant cell. We have nuclear membrane, Golgi apparatus, nucleus, cytoplasm, starch granule, smooth endoplasmic reticulum, and the rough endoplasmic reticulum, chloroplast, centrosome, cytokeraton, ribosomes, cellulose, cell wall, cell membrane, vacuole. Then, the animal cell. We have nucleus, nucleolus, nuclear membrane, cytoplasm, centrosome, ribosome, rough endoplasmic reticulum, pinocytic vesicle, lysosome, fat droplets, glycogen granule. Then the Golgi apparatus, mitochondrion. Then the last is the smooth endoplasmic reticulum.
last is the structure of animal cell and plant cell. First structure is the mitochondria. Animal cell is also present in plant cell. Next is cell wall. Animal cell is none, plant cell is yes. Plastids. Animal cell, no. And then plant cell, yes. Vacuole. The animal cell is one or more small vacuoles, much smaller than plant cells. Next, the plant cell is a large central vacuole taking up 90% of cell volume. So ribosomes. Animal cell is present, then the plant cell is also present. Centrioles. Animal cell is always present, and the plant cell is only present in lower plant forms. Lysosomes. In the animal cell, lysosomes occur in cytoplasm, while plant cell, lysosomes usually not evident. Next is plasma membrane. In animal cell is present. In plant cell is also present. The cell wall in animal cell is absent, while plant cell is present. The chloroplast in animal cell is absent, while plant cell is present. Last structure is the nucleus. An animal cell is present and the plant cell is also present.
So that's all of our discussion. I hope you learned something of our topic about the types of the cell structure, and function. That's all.